Alright, this is the topic of genetic engineering, which is actually a technique where we transfer gene or genetic material from one organism to another. It has several applications, which includes the gene transfer of humans to a bacterium while producing insulin. Genetic engineering can also be used to transfer genes from one plant species to another. The crops that are being produced as a result of it are known as genetically modified crops. Genetic engineering has also played a vital role in the treatment of certain diseases, for example, cystic fibrosis. Let's see the production of insulin in detail, which includes several steps. Step 1 involves the isolation of human insulin gene from the nucleus of a cell from a healthy person. This is done by use of a restriction enzyme. As you can see in the diagram here, a human cell is taken and that gene is being isolated, which is responsible for the formation of insulin in humans. This is done by the use of restriction enzyme, which is able to cut the DNA at the required points. Step 2 involves the use of restriction enzyme again, but this time it is used to cut open the bacterial plasmid. From your previous knowledge, I hope you know that a bacterial cell contains additional rings of DNA, which is known as plasmid. As you can see in the diagram that here the plasmid is being cut open, and this is again done by the restriction enzyme. Step 3 involves the use of another enzyme known as ligase. This is used to join the human gene with the plasmid DNA to form a recombinant plasmid. This can be understood by looking at the diagram. If you can see that the shaded part is showing you the isolated insulin gene, and now we have pasted it or inserted it into the bacterial plasmid. This plasmid that is being formed is known as recombinant plasmid. This will be introduced to the bacterium again. The bacterium which is produced as a result of it is known as transgenic organism. This is any organism which would carry the foreign gene. In step 4, we will make sure that we provide suitable culture medium, temperature, pH and oxygen to the bacterium which we have produced, that transgenic organism. This bacterium will now reproduce by binary fission, which is an example of mitosis. We already know that in mitosis, the parent cell produces identical copy of itself. There is no genetic variation between the parent cell and the daughter cells. As you can see that the bacteria has reproduced, and as a result of it, it has produced multiple bacterium cells which are actually identical copy of the parent cell. My desired gene, which is the human insulin gene, has passed on exactly like that into the bacterium cells which are being produced, and obviously all these cells will produce, as a result they will produce insulin just like their parent cell. At the end, we will make sure that we extract the desired product, which in this case is insulin. After extraction, we will purify the insulin, make sure that it is being separated from the bacterial colony, and we can use it in diabetic patients. The next diagram shows you the summary of all these steps together. Again, I'm repeating that the human cell would contain a gene which is responsible for formation of human insulin. That gene will be isolated and will be inserted into the plasmid of the bacteria. Once the plasmid is being formed, this will again be inserted into the bacteria. This bacteria will now then be allowed to reproduce, making sure that this bacteria which is going to reproduce has appropriate temperature, oxygen, culture medium and pH. The bacterial cells which are being produced are as a result of mitosis, binary fission or asexual reproduction, resulting into identical copies of the bacterial cells. Each and every cell which is being produced is now able to produce insulin, as all of them have the human insulin gene.